And unplugging it. Okay. Oh. Great. Well, thank you so much, Marty, for the invitation, and Karsten. It's really great to be here. It's weird to be down here. And I realize I'm standing between you and beers, and hopefully I'll be able to convince you that resonance Raman spectroscopy is cool enough to wait for beers. It's definitely one of my favorite spectroscopic techniques, and it's incredibly useful. I wanted to give you first some basics on resonance Raman spectroscopy, some instrumentation information, and then also walk you through some of the work that my lab has done using resonance Raman spectroscopy to characterize different metalloprotein systems. So fundamentally, resonance Raman spectroscopy is a vibrational technique, which means it's a structural technique. And we can get out all of the same information that we can, or actually more information, as any other vibrational technique, often complementary information, where we can learn about the active site structure. We can use different isotopes to learn about the nature of the atoms involved in the bonding. We can also learn about symmetry. It's complementary to IR spectroscopy, and so modes that are enhanced in IR spectroscopy are often not seen in Raman, and vice versa. Because it is so diagnostic, we can look at characteristic frequencies to tell us about different functional groups that are in our system, and we can also look at changes across a series to see, as we modify one thing, how those frequencies change and how that may report on structure. But beyond being a geometric technique, resonance Raman spectroscopy also provides us information about electronic structure. Because of that whole resonance component, we can learn about the potential energy surfaces, but we can also learn about excited states, the energies of the excited states, the nature of the excited states, and we can actually quantitatively map out the essentially geometry of the excited state, which is very relevant in considering things like charge transfer and modes that are coupled to charge transfer. Additionally, resonance Raman spectroscopy has inherently very high time resolution, which means we can do all of the same fun techniques, freeze quenching techniques, rapid mixing techniques, and trap different intermediates, looking at how reactions progress over time and how structures change over time using different apparati. And so we can learn quite a bit about structures of different systems coming from different viewpoints. So fundamentally, because resonance Raman spectroscopy is a vibrational technique, we look at vibrational frequencies, and already the frequencies themselves tell us quite a bit about the bonding between the atoms and really the identity of the atoms. And just for some basics, the vibrational frequency is directly related to both of these, the potential energy surface and those atoms, where the frequency is directly proportional to the square root of the force constant over the reduced mass. And so this is really the root of the isotope shifts when we use isotope shifts to look at what modes we actually have. And just to give you an example of that, one of the largest isotope shifts that we actually see is for HD exchange. And so if we look at the ratio of the reduced masses, we typically assume that the force constants are the same as we change isotopes. And generally speaking, that assumption is fairly valid, because force constants, as we'll see, just derive from the potential energy surface. And so if we make that assumption, then we can see that the ratio of the frequencies between something like HCl and DCl should be about 1.4, and that's because it's directly proportional to the inverse of the reduced mass. So we see very dramatic shifts for HD exchange. Smaller shifts are often seen for heavier isotopes, C13, O18, and if you do the resonance Raman practical, you'll see these shifts, but while they're smaller, they're often very noticeable and very pronounced, and that can tell us a lot about what the structure of those active sites are. We can also use vibrational frequencies to tell us about the potential energy surface. Here I'm showing just a standard Morse potential, and if we consider what vibrational frequencies really are and what they actually report on, the force constant is really just the second derivative of the potential energy surface, essentially evaluated at the equilibrium geometry. And so the force constant reports on the curvature of that potential energy surface, which means it tells us about bonding. It tells us about the dissociation energy, and it tells us about the steepness of that curve, which changes depending on the order of the bonds. So bonds that are double bonds are much steeper than single bonds, and so on. And so putting these together, just by looking at the frequencies, you can already tell quite a lot about the system that you're looking at. Because of this, there are some characteristic vibrational frequencies that actually occur in resonance Raman spectroscopy that mirror some of those that are seen in IR spectroscopy. So I'm sure many of you are aware of sort of our canonical IR organic functional groups that we see in IR. I've just listed some sort of more traditional frequency ranges here. But in bioinorganic chemistry, particularly in Raman spectroscopy, we also need to consider characteristic vibrational frequencies of amino acids. So I've shown a few of the amino acids that are typically involved in coordinating metal centers and characteristic frequencies that are associated with them. So cysteine is certainly an amino acid we care a lot about. We always see the CS bond, and you can sometimes see that SH bond, which are fairly unique frequencies relative to most organic moieties. Histidine comes up a lot, and then tryptophan, tyrosine, and phenylalanine are aromatic, and we'll talk about selection rules in a minute. But those can actually be very useful to use as internal standards, internal reporters for intensity analyses and normalization. And so when you see these bands in your resonance Raman spectra, they may not actually all be resonantly enhanced bands. You do have to consider that there are Raman scatterers, and often they're present in fairly high effective concentrations in your protein when looking at these spectra. Additionally, characteristic or sort of traditional metal ligand motifs have very characteristic frequencies. So 4-iron, 4-sulfur clusters have been fairly well studied. Depending on the oxidation state, they have different frequencies, and they have a very different fingerprint than, say, 2-iron, 2-sulfur clusters or 3-iron, 4-sulfur clusters. So you can actually use these frequencies as both oxidation and structural markers. Metal thiolate bonds typically stretching vibrations typically occur between 300 and 400 wave numbers. Metal oxo and metal hydroxo bridging species actually tend to occur a bit higher in frequency for both the asymmetric and the symmetric stretch. And then porphyrins, which we've heard a lot about today, have of course very diagnostic vibrational modes. The in-plane stretching mode is really pronounced, and there are vibrational markers both for spin state and oxidation state that you can use to really learn about the nature of the iron center, as well as sometimes even identify the axial ligands coordinated to it to look at structure and small molecules that are bound to porphyrins. In addition to looking at sort of geometric structure, we can use resonance Raman spectroscopy to look at electronic structure. So if we think about selection rules, traditional IR spectroscopy is a very conventional technique. It really is just the excitation from the ground vibrational state into an excited vibrational state, and that has IR energies. The intensities of IR transitions are directly proportional to the change in dipole moment along the change in coordinate. And so for a molecule, for a vibration that has a large dipole moment change, those vibrations are very strongly enhanced in IR spectroscopy. In Raman spectroscopy, traditional Raman spectroscopy, we populate what's called, or typically referred to as a virtual state, where really it's the electric field of incoming light actually interacts with the electric field of our molecule. And that induces essentially a scattering event, or really a photon scatters off of that. It can either scatter elastically, and that's what gives rise to Rayleigh scattering, or it can scatter off of the molecule inelastically, depositing energy into a vibrational quantum of the molecule or the protein system. And so in that case, the light that you'll see will actually be shifted from the incident light by exactly the energy of one vibrational quantum. And so it gives you complementary information, but in this case, the intensities are not proportional to the dipole change. They're actually proportional to the change in polarizability along that normal coordinate. And so molecules that are vibrational modes that show a large change in polarizability, in particular molecules that have a large degree of polarizability, things like benzene rings, tryptophan, and tyrosine, those in-plane stretching modes change the polarizability of the molecule quite a bit, and therefore they're very good Raman scatterers, which is why they often show up in spectra of proteins. In resonance Raman spectroscopy, the intensities become a bit more complicated, because now we directly populate, or we use an excitation wavelength that's resonant with an electronic excited state of the molecule. That can then interact and inelastically interact, scattering off a photon that's different by the same energy, but now the intensities report not just on the polarizability change, they actually report on the overlap between the ground and excited vibrational and electronic states. Additionally, resonance Raman intensities have this resonance enhancement term down in the denominator, and so as your excitation wavelength starts to approach the electronic transition, that term gets very small, the intensities get very large, and this gives you the selectivity that resonance Raman spectroscopy provides over just simple Raman spectroscopy or IR spectroscopy. And so there's really two components that go into these enhancement factors here, where resonance enhancement can actually give you intensities, they say, that are up to 1,000,000-fold greater than Raman scattering. Traditionally, it's more like 1,000 to 10,000, but they can be incredibly strong depending on the system. The key here is that modes that have a very large displacement in the excited state are the most intense modes. To understand that a bit further, we can look at actually the equations dictating resonance Raman intensities. There are actually two formalisms for resonance Raman intensities that were developed. This sum over states expression is what I showed on the other slide. This is that proportionality factor that I showed, where we look at the overlap and then the resonance enhancement term. The other key factor to keep in mind here is this e to the s cubed EL, which is really the energies of the incoming and the scattered photon. These essentially get lumped together to give you approximately an energy to the fourth scaling, which means Raman scattering, actually all scattering, scales as nu to the fourth. So at higher energies, you get greater scattering efficiency across the board. This is important to keep in mind when selecting an excitation wavelength. The other formalism that is actually a little more simple to evaluate numerically is actually this time-dependent formalism, where we think about our wave packet along the ground state being vertically projected up to the excited state and then propagating along that excited state in a time-dependent fashion. And really the rate at which it propagates along is proportional to the steepness of the excited state, and that steepness is really dictated by how displaced that excited state is relative to the ground state. So at the end of the day, these give the same answers. They're quantitatively the same expression. We end up seeing that resonance Raman intensities are proportional to the frequency of the vibration squared and this dimensionless displacement delta squared. And so when we think about quantitatively evaluating resonance Raman intensities, we can actually look at where this excited state is relative to the ground state. And basically said that the greater the displacement, the larger the intensities are. The flip side is that if the geometry in the excited state is not that different than the geometry in the ground state, for example, D to D transitions, you really don't have any resonance enhancement, even if you have quite a large extinction coefficient. And so both of those things play into dictating whether you will see a very strong resonance Raman signal. The other thing that I want to mention is that you can actually, because these are numerical equations that you can actually solve, you can quantitatively model resonance Raman spectra in conjunction with absorption spectra to actually get a number for this delta, and correlating that back to the nature of the mode, you can actually get the distortion in the excited state. And that process is generally done iteratively. It can be a very long process, but actually you can get quite a bit of information out about reorganization energies for charge transfer transitions and so on. So if you'd like to quantitatively model a resonance Raman spectra, I'd be happy to talk more about that a bit later. It is kind of a long process, but it's really fun. I did a lot of that in graduate school. So, in addition to looking at electronic structure, resonance Raman spectroscopy can also be used to look at reactivity. And so inherently, as I mentioned, resonance Raman spectroscopy is very fast time resolution. You can do rapid freeze quenching experiments and quench your samples down to approximately 10 milliseconds. The key with RFQ and Raman, or resonance Raman, is that you have to use liquid ethane. You can't use isopropanol because isopropanol or isopentane is such a great Raman scatter. But if you use liquid ethane, you can actually do RFQ resonance Raman. You can do rapid mixing and continuous flow resonance Raman. You can also look at very fast processes, photoinduced processes, down to the picosecond or so. And so photoinduced isomerization processes, and spatially separating the pump and probe is useful if you're doing photoinduced processes that then cascade down to reactivity. And so a setup that we've built to actually do this is a rapid mixing device where we use, attached to here are two different syringes, and then there's a quartz capillary that you can kind of barely see. You can probably see it better in your slides. Where we actually probe the sample. This is about 100 micron capillary, and we can actually use a second light source coming in ahead of the probe site to separate the pump and probe spatially, which means the fluorescence that we'll see really won't interfere with the Raman scattering. And in this case, we did this on a microscope-based system to really focus both the pump and the probe, and we use this system to look actually at an oxidation reaction before deprotonation. And so this is the oxidation of tryptophan using cerium. We were looking for this tryptophan cation radical before deprotonation, and by varying the flow rate, you can essentially vary your time delay and follow the peaks as they grow in and decay. So with that, I want to transition into talking about some instrumentation and instrumentation constraints in resonance Raman spectroscopy. You really only need a few different things to do resonance Raman spectroscopy, and I know everyone thinks that it's really very simple. You just have to buy a few different things. With resonance Raman, you need an excitation source. You need a light source. As I mentioned, scattering efficiency goes as nu to the fourth. You want a monochromatic light source. You want it to be coherent. And you want polarization control. And so while C.V. Raman actually did his experiments with a mercury lamp and a polarizer, it's much better and easier these days to do your experiments with a laser. You also need narrow bandwidth. And while I mentioned you can get very fast time resolution, that's actually limited by the Heisenberg uncertainty principle. And so pulses that are about 100 femtoseconds end up being about 150 wave numbers broad. That's much broader than most Raman lines. And so you really have to take that into consideration. Using ultra-fast lasers for resonance Raman or Raman spectroscopy is really not tractable. But if you drop that by a couple orders of magnitude, now your line widths are within something tractable. You also want to make sure that you have variable power. Going up to higher powers, signal scales linearly with power, but you can often induce nonlinear processes, two-photon processes. So being able to vary the power systematically and make sure that the signal does indeed scale linearly with power is a control experiment that perhaps is one of my pet peeves with resonance Raman spectroscopy not everybody actually shows. After you have your excitation source, you need a sample. Easier said than done. You need a sample that actually absorbs at a wavelength that you can access. So you have an electronic transition within an accessible wavelength, reasonable concentrations. You can do resonance Raman spectroscopy because of this resonance enhancement down to tens of micromolar. Ideally, things typically work better at hundreds of micromolar, although if you do have a highly scattering system, a porphyrin or a coronoid, 10 micromolar is typically used. One thing to consider if you have something like an iron-sulfur cluster or de-enhancement mechanisms, that's another talk altogether. If you're interested, I'd be happy to talk to you about it later. But iron-sulfur clusters and iron-oxo clusters will often have lower resonance Raman intensities than you think they should have based on the extinction coefficient. And that's because of these de-enhancement mechanisms. So if that's what's going on in your sample, that may be something to think about. Fluorescence. Fluorescence is really the biggest enemy of resonance Raman spectroscopy because fluorescence obscures all of those Raman scattered bands. And so it's important that your sample is either non-emissive or that you're able to probe blue of the emission and take advantage of Kasha's rule. You also need to make sure you have a very clean sample and that you don't have fluorescent contaminants. Oftentimes our proteins look very clean on a gel, but NAD or FAD containing proteins can be fluorescent, and so sometimes those contaminants also interfere with Raman spectra. Additionally, photochemistry, if there is photochemistry happening at the probe wavelength, that's something to really check for. This is where that variable power comes in. The last thing that you need is some way to collect those scattered photons. So we need, because those scattered photons are fairly few and far between, you need a very high efficiency collection lens. We need to disperse the photons, typically a monochromator is used, and then you have to turn those photons into signal, and so either a PMT or a CCD is used. So as a sort of depiction of a very minimalistic resonance Raman setup, you have a laser, you have a sample, we collect the scattered photons and then focus the scattered photons onto the entrance of a spectrograph where they're then dispersed and imaged on a CCD. In my lab, we have a somewhat different system that I just wanted to spend a few minutes talking about because while traditional lasers are typically ion lasers are used, and nowadays actually diode lasers are being used more and more for Raman spectroscopy, another approach is to actually use pulsed lasers, but use them operating in a quasi-CW mode. And so we actually have a pulsed picosecond tsunami laser. It's a titanium sapphire laser, a very tunable wavelength, and tunable excitation lengths, which gives us bandwidths that are narrow enough to actually still see the Raman lines and resolve the Raman lines, but have a peak intensity so that we can actually do nonlinear nonlinear spectroscopy actually with the laser itself. So now we can generate the second harmonic, the third harmonic, the fourth harmonic, and even some frequency generation using this system, which effectively gives us continuously tunable excitation from the deep UV all the way into the near IR. We've also coupled this with a single monochromator system. So this is actually, often people will use triple monochromators. In our lab, we use a single monochromator, which gives us high throughput. For Rayleigh rejection, in the case that we don't have a convenient line, we use a prism pre-filter, which has, again, high throughput, but dispersion, enough dispersion that we can actually separate the Rayleigh line from the incident beam. And so this allows us to look globally at the entire structure of the protein. We can look at aromatic amino acids using the fourth harmonic, histidine, tryptophan, tyrosine. In the deep UV, we can actually look at protein structure, global protein structure, secondary structure, and structural changes that occur because we're resonant with the amide backbone. Going a bit redder, we now have sulfur excitations that we can start to probe. Thiolane disulfide bonds, we can look at nucleotides, and then in the visible are the transitions that we're all more familiar with, the metal to ligand charge transfer transitions, the porphyrin pi to pi star transitions. This gives us information about bonding. And in the IR, we can start looking at intervalence charge transfer bands and radical states, which again give us a lot of information about bonding and electronic configuration, as you heard. In full disclosure, the disadvantage of this system is that while it's a very useful system and very versatile, picosecond titanium sapphire lasers are not as common and therefore not as stable as femtosecond lasers, and so they are somewhat sensitive to drastic humidity and temperature changes. So that's something to keep in mind because they are pulsed and the pulses come not from, or the pulse length comes not from a high reflectance mirror, but actually an interferometer optic. To show you what this looks like, this is the output of our TISAF. We have the red light comes out, it's focused into a second harmonic crystal, which then generates the blue light as well as the red light. We then dump the red light and send the blue light all the way down to the sample. And it actually looks quite pretty. In the dark, you can see the crystal glow. Once it goes to the sample, and I just want to show you a few different configurations, you'll see a slightly different configuration as well if you do the practical. But once it goes to the sample, the light comes in, and this is an EPR tube in backscattering geometry, where it actually comes in and then is collected, hits the sample, is collected by the collection lens. The collection lens collimates it to this focusing lens, which then focuses it onto the spectrograph. A slightly different configuration is the right angle configuration. This is a fairly simple one to set up. That just really looks, the beam comes in and then is bounced up to the sample, and then again the light is collected by the collection lens here. Another type of sample configuration that we use and that many people use is a low temperature configuration because this allows us to look at quenched samples. It also allows us to avoid photochemistry. And we just use a finger doer here. Oftentimes you can use a cryostat if you need to get lower than liquid nitrogen temperatures. You have to use a cryostat. But this is a compromise between simplicity and interchangeability, and one that you might consider doing if a cryostat isn't easily accessible. And you can see the backscattering geometry a bit easier here. The light hits this mirror, is focused by this lens, and then hits this tiny mirror before it goes up to the sample, and then again the light is collected. So these are the different sample holders and configurations that we have, and then you'll see a continuous flow setup as well during the practical. So in the last few minutes, I want to tell you about how we've used these systems to actually characterize and understand the structures of a couple different protein systems that we work on in our lab. The first is a structural study of the protein azurin, and azurin's been used a lot in this today as a case study. It's actually a very fantastic case study. It's a well-studied protein. We've talked a lot about the cupridoxin, the type 1 cupridoxin coordination motif. There's a nice absorption band. What we haven't talked about is this tryptophan residue. Azurin has a very hydrophobic tryptophan residue and this nice beta barrel fold. So we can use this variable excitation resonance Raman spectroscopy to excite not just the copper center, but also the tryptophan and the amide backbone. And what we see are very different excitation profiles, very different spectra, depending on where we excite. In the very, very deep UV, we really just see bands corresponding to the amide bands. And the position and intensities of those bands actually report directly on the secondary structure. And you can use this to actually pull out the percentage of beta sheet relative to alpha helix and actually look at structure, which of course is useful if you don't have a crystal structure of the protein. The tryptophan residue has very nice pronounced peaks. You barely see it at 206, but 228 is an optimal wavelength for that. And the frequencies and intensities of these peaks that I've indicated here all report on the nature of the tryptophan. And so you can see that we have a very hydrophobic tryptophan that's not involved in any cation-pi interactions. And we can actually get orientation and geometry from the frequencies of these W3 and W7 markers. The copper center has a very pronounced metal thiolate vibrational mode here, actually at fairly high frequencies, at 408 wave numbers, which is much higher than typical metal thiolates are typically below 400 wave numbers, but this is a fairly unique system in terms of that bond strength. You can also see the copper histidine modes, and at higher frequencies you can see this carbon-sulfur stretching mode that inversely correlates with the bond strength of the metal thiolate. So the carbon-sulfur mode gets higher frequencies as the copper-sulfur mode gets lower. So we can learn a lot about structure from this system. With this, we could also look at changes during reactions, changes in global structure, any involvement of aromatic amino acids, and then changes in the active site. We can also use this to look at how this protein structure changes as we change the protein a bit. And so we do quite a lot in my group with nickel. We can take copper azure and actually put nickel in. We see it changes the absorption spectrum quite a bit. We can then take that nickel azure and mutate it, changing one of the methionine ligands to an alanine, and look at how that changes the structure of the active site. What we see if we use these different excitation wavelengths, essentially exciting into the same part of each transition, is that this metal-sulfur band actually shifts sequentially to lower frequencies, indicating a decreased metal thiolate bonding interaction consistent with what we might expect going from sort of the optimized system to a fairly perturbed system. This carbon-sulfur bond goes up in energy really accordingly with that decrease in metal thiolate bonding. The other thing that we notice is a very different intensity pattern. In these high frequency regions, we now see fairly intense modes. This can report on changes in the amide configuration, but it can also report on histidine. And actually, this is a characteristic frequency for histidine, where the nature of the electronic transition may have changed and may be delocalized now more onto the histidine. And so in addition to looking at geometric structure again, we can start looking at these electronic transitions. Another system, and the changes are, we see the changes essentially recapitulated if we do calculations on the structures of these systems. That bond length gets longer, going from copper to nickel, and then the geometry changes quite a bit as we change the methionine to an alanine, and you can see that histidine geometry change. In addition to looking at adjuvant structure, we also look at nickel rubidoxin. We use nickel rubidoxin as a model for hydrogenase proteins, and in trying to understand how that works, we want to be able to look at the structure of these different intermediates. And this is work by Jeff Slater, who's here helping you learn resonance Raman spectroscopy. So if you have any questions, definitely try to find Jeff this week. But we essentially drew out a mechanism for nickel rubidoxin where we go through this protonated thiolate intermediate, a metal hydride, a thiolate hydride, bis-thiolate hydride species, and thought, well, a lot of these species should be Raman active. Metal hydrides have very nice vibrational modes. We should be able to see this thiolate. How do we do that? Well, before we could do that, we needed to make sure we understood the resting state of the system. So we took the nickel rubidoxin and actually did resonance Raman spectroscopy across the two charge transfer bands. Nickel rubidoxin has two visible charge transfer bands, or really one in the visible, one in the near UV. And we excited both at the peak and about halfway down each of those transitions to really characterize the resting state of this catalyst. When we did that, we saw very different intensity profiles as we moved through the excitations. So in the near UV transition, we see these three bands very pronounced. In the visible transition, now the intensity pattern has changed completely, and we see fairly high intensity bands in the high frequency regions. So this wasn't necessarily what we expected to see. We did normalize these to the phenylalanine, as I mentioned. The band positions are identical moving across there, and that's something that's important to point out, that the vibrational frequencies shouldn't change if you change excitation wavelengths, just the intensities. So we took this data and wanted to actually get a bit more out of it. We wanted to understand why we saw these high frequency bands enhance so much, if we could correlate this to structure and structural changes, and if we could actually learn a bit more detailed information about the electronic structure. And so for this, we actually turned to DFT calculations to really quantitate and quantify our resonance Raman intensities and correlate them. To do this, however, we needed to figure out how really how to do the DFT calculations, how to pare down the active site to a model that we actually could calculate. And so because this is something that really, because we can't actually calculate the full protein is quite large, and while it is small for a protein, it's much too large to calculate quantum chemically. We actually considered 3 different models. The very simplistic nickel-NACA model, where we just use N-acetylated C-terminal amidated cysteine, 4 of those. We created what we call a backbone model, where we had the CXXC loops coordinating coordinating the metal center, but had the side chains replaced with glycines. And then we did the full model, where we had the whole secondary coordination sphere with those full side chains included, and calculated the structures of all of these. We also had to make sure that the electronic structures match. So we used TD-DFT calculations to calculate the absorption spectra, and already we can see that the nickel-NACA simple model really doesn't work. The absorption spectra really don't reproduce either the frequencies or the intensities of the transitions, and we really just have this large, broad distribution. The backbone model isn't bad. We are able to get the energies a bit better, but this sulfur transition is actually split into 2, and really we actually found that this full model was what we actually needed to use to get, or to perform, to advance with the calculations. So we started with this full model, and we took these 2 different transitions, and we used structural validation against a crystal structure of nickel rubidoxine, and then we calculated the resonance Raman spectra. We did this using the ORCA program, actually the ORCA-ASA module in the program, which actually relies not on geometry optimizations of the excited state, but actually the slope of the excited state. And so I mentioned that if you know what the slope is and you assume a harmonic potential energy surface, you can get this displacement here. So that's basically what the ORCA program does. It's computationally very tractable, even for these very large systems. And what it does is maps out the displacement along each coordinate for a given excited state. So to use this, it's quite straightforward to use. You simply specify the electronic transition that you're interested in probing and the excitation energy. It's important to consider this relative to the TD-DFT transition energy. And so if your energies are all shifted, here actually we didn't apply any sort of energy offset. So you can see the D to D transitions are reproduced, as well as the 2 sulfur to nickel charge transfer bands. But if you do apply an energy offset, which is fairly common with TD-DFT, you have to offset your excitation energies as well. And then this ORCA-ASA program takes these displacements and can actually map onto a resonance Raman spectrum. And what we saw was actually strikingly good agreement between the experimental and the calculated spectrum. We took this high energy state, which represents a sulfur P sigma to nickel charge transfer transition. Just looking at the nature of the state, we'd expect our totally symmetric modes to be enhanced, and that is essentially exactly what we saw for excitation into this transition. The lower energy visible transition is a sulfur P pi to nickel charge transfer transition. So looking at the symmetry of this mode, we would expect the asymmetric modes to be enhanced. That again is what we see at the lower frequencies. And so we were actually quite excited that this worked so well, both in the frequency and in the intensity space. And so we can now use this to actually do normal mode assignments. The gold standard for normal mode assignments is selective isotopic exchange, and doing that essentially 1 by 1 to really identify which modes are involved in a transition. However, we all know that doing that for proteins can actually be impossible, if not very expensive and fairly intractable. But now that we have good calculated resonance Raman spectra, we can actually use these calculated spectra to identify the modes and assign them to experimental transitions. And so these are now normalized to the maximum intensity, and again you see very good agreement across this whole spectral range. We can identify the 276 wave number band as the totally symmetric mode, and these 2 flanking bands as the asymmetric modes. The other thing that's actually really cool is that we can pull out 2 distinct carbon-sulfur stretching modes. And this is a high-spin tetrahedral nickel-2, which isn't perfectly tetrahedral. In fact, it's distorted because of Jahn-Teller distortion to 2 long nickel-sulfur bonds and 2 short nickel-sulfur bonds within an approximately D2D symmetry. And we actually see that not just in the calculations, but we can actually start to pull out different carbon-sulfur stretching modes in the vibrational spectrum. And so we can use that moving forward because we've been able to identify these bands and see how they change as we change catalysis. The other thing that I wanted to point out is that these high intensity, high frequency bands that we see at longer wavelengths were really only enhanced for the full model. The backbone model didn't show the same level of enhancement. So now that we have the electronic structure of the resting state characterized, we can now move forward and actually start characterizing these intermediates, looking for changes again in those carbon cysteine modes, as well as metal hydride modes. And so that really represents a lot of our ongoing efforts in the lab on this project. In the last couple minutes, I just want to tell you, sort of go the next step from geometric to electronic structure to reactivity, and tell you a little vignette about when you don't get exactly what you expect from resonance Raman spectroscopy. So this is a project on the R2-LOX protein that we work on in my group. This is a heterobimetallic manganese iron protein, and we wanted to understand the structure at a very detailed level, both for the manganese iron and for the di-iron loaded protein. So we looked at the absorption spectrum of R2-LOX relative to the apoprotein. We saw the characteristic iron-oxo charge transfer bands. There was a bit of a tail out to the visible region, and so we decided to go pre-resonant of the electronic transition and start looking at the vibrational spectra as a marker for structure. The apoprotein had essentially no bands that were really particularly pronounced. You can actually see the tryptophan and tyrosine and phenylalanine modes here. You can see the amide bands there, but these are Raman modes, not resonantly enhanced modes. When we looked at the metal-loaded hollow proteins, we now see a lot of bands. We see bands in the low frequency region, which is what we expect to see. Metal ligand bands should have fairly low energies, but we saw these very pronounced bands at high frequencies. Much more pronounced and much higher frequency than we necessarily would have expected to see. This was very puzzling to us, but going back through the literature and thinking through what we know about those bands, as well as what we know about different classes of metalloenzyme, we realized we pulled up old resonance Raman spectra of the purple acid phosphatase proteins. And we noticed that the purple acid phosphatase proteins all have this characteristic quartet at essentially identical vibrational frequencies to what we saw in R2-LOX. The purple acid phosphatases are purple because they have a metal tyrosinate coordination. And so we started to think about whether R2-LOX could also have this tyrosinate coordination. The structure's been solved. We know that in the resting state it doesn't have this tyrosinate coordination, but seeing this in resonance Raman led us to do some photochemistry experiments. The protein is clear, but upon irradiation it actually does turn bright purple. We've characterized that now quite a bit further, looking at the change in absorption spectrum. It has a fairly high quantum yield. We were able to get a structure of that purple species, and we see that indeed this glutamate residue is actually decarboxylated, and we have direct tyrosine coordination of one of the iron centers. So what this showed us was that we do have photoinduced reactivity in the system. This gets back to that photochemistry that I was talking about. You wouldn't necessarily expect it. There was really no indication that we should have this photochemistry, and it's very well-defined photochemistry. It actually only decarboxylates at one of the glutamate residues, that really occurs, we think, selectively because R2-LOX has a bridging hydroxide ligand rather than a bridging oxygen ligand. So we would have completely overlooked that, though. We never really would have identified this purple conversion if the resonance Raman spectra hadn't been so distinct and so different from what we expect for this class of species. So I hope that I've been able to show you some examples and convince you that resonance Raman spectroscopy can be applied actually broadly to a lot of different systems. You can use it to look at geometric structure and electronic structure, as well as different kinds of reactivity that occur. And hopefully if you do the resonance Raman practical, you'll get a better sense for hands-on how to actually use resonance Raman instrumentation. So with that, I'd be happy to take questions.